Well, thank you very much for that nice introduction, Gary, and thank you all for coming this afternoon. I was going to say you can't underestimate the benefits of a free sandwich, but I don't think there were any free sandwiches. There might be that at the end. What I'm going to concentrate on this afternoon, I'm going to concentrate on two aspects of work that I've been doing during my time here at Heriot-Watt. The first relates to reservoir engineering, so that's looking at multi-phase flow through porous media. And the second is an adaptation of the modelling methods I've been using in that subject area to look at blood flow and capillary growth in the context of tumour growth, wound healing, and the developing retina. So before I start, no research is done in isolation, and I have to thank all of these people without whom I wouldn't be here this evening. First and foremost, Ken Sorby, who's here, who gave me my first job here 28.5 years ago, gulp. And as you said, it's the same slide background as we used 28.5 years ago. Mark Chaplin, who's at St Andrews, was in Dundee. I've done a lot of work with Mark on the biological modelling side of things, specifically angiogenesis and tumour growth. Sandy Anderson, who's now in Florida, but worked with myself and Mark during the early stages of the biological work. Jonathan Sherritt from the Maths department here, without whom the second half of this presentation wouldn't exist. He put me in touch with Mark and Sandy up in Dundee. It was a complete fluke that we actually got together and started doing this sort of work. Angelique Stefanou, who's now in Grenoble, she was one of my RAs who helped with the angiogenesis, adaptive angiogenesis work, which I'll talk about later. Chris Mitchell and Andrea Devlin, who are both at the University of Coleraine, who worked with us on the retinal modelling, and they did the experiments, the mouse experiments. Vasilis Boros, who's got me interested in contrast-enhanced ultrasound, and we have a joint PhD student, Vaso, who's sitting over there, and Jayashree Kalpathy-Kramer at Harvard, who I've been working with, looking at brain imaging, looking at tumours, and modelling multi-phase, modelling tracer flow through mouse vasculatures. And also my PhD students, some of whom are here today. And again, a lot of the work you'll see this afternoon has been done by my PhD students. So without them, a lot of the work that I'm about to describe wouldn't be up here. So a brief outline, I'm going to start by talking about a very simple flow law, the Hagen-Poiseuille flow law, which gives an equation for the volumetric flow rate going through a simple cylindrical capillary tube. I'm then going to give a very brief summary of reservoir engineering, and I'm going to describe how we can take that simple tube to build it into an interconnected network and then apply that interconnected network to the study of multi-phase flow through rocks, so different fluids flowing through rocks. And I'll briefly touch upon a numerical simulator that we've developed called Numbskull, which allows us to do lots of little numerical experiments covering a wide range of different petrophysical problems. I'll then go on to adapt these pore network modelling algorithms to the study of capillary growth and blood flow in tissues. So we've gone from tubes to rocks, we'll then go from rocks to tissues, and specifically I'll talk about something called angiogenesis, which is the growth of new blood vessels from pre-existing vasculature. And I'll touch upon another little simulator called NUMTI, which is sort of the sister code to Numbskull, which allows us to look at lots of different biological processes, wound healing, retinal development, ultrasound, etc. And then I'll finish by talking about the current challenges that I'm interested in and some future opportunities. So it all begins with these two chaps, Godhilf Hagen and Jean Poiseuille, one Prussian and one French, and there's not a great deal known about these two guys. Hagen married his niece and had 7 children, which is probably the most interesting thing about him. And other than having a bit of a Napoleon complex, Poiseuille also invented the U-tube manometer. Other than that, there's not a great deal known about them. But what we do know about them is that they were both very interested in solving the problem of fluid flow in pipes. And Hagen did a number of experiments and came up with this flow law that the volumetric flow rate going through this simple cylinder was approximately proportional to the radius of that cylinder to the 4th power. And Poiseuille did a more extensive range of experiments and found that the flow rate was related to various parameters via this equation. So if we double the viscosity of the fluid, we'll halve the flow rate through the tube. If we double the length, we'll halve the flow rate. If we double the radius, the flow rate will go up by a factor of 16. And that's very important when we're interested in blood flow, because a very small change in the radius of a blood vessel can have a big impact on the delivery of blood and nutrients to tissue. So that is known as the Hagen-Poiseuille law, and unfortunately for Herr Hagen, it's really referred to as Poiseuille's law in day-to-day. I just call it Poiseuille's law. So Hagen's kind of dropped off, but I wanted to acknowledge his contribution to the derivation of the law here. So we've looked at a simple tube. Now how can we adapt that simple tube model to study the flow of multiple fluids through porous media? So an oil reservoir, unlike what its name might suggest, is not a big lake of oil underground. It's essentially a cliff. I'm waiting for my petroleum engineering colleagues to fall off their seats when I give this very brief introduction to this. It's essentially a cliff buried thousands of feet underground, and that rock acts like a big sponge. And so the oil and water and possibly some gas is trapped within the pore spaces between the sand grains that make up that rock. And the geology can be incredibly complex. So you've got lots of different structures at different length scales, and it's quite a tricky problem to try and model flow through that. And petroleum engineering is all about trying to optimize the flow of oil from different types of reservoir. And so we can map these reservoirs, and we can populate a numerical model with lots of different blocks, and they each have different flow properties, different permeabilities, different porosities, and so on. And say in the North Sea, where this might be full of oil, we could put an injector well here, and we could inject seawater and flush out this formation and produce the oil out here. But solving that flow problem is really quite difficult. So we divide the domain into lots of different blocks, and we have flow laws for different fluids going in and out of that block. And so this is just a conservation of mass equation for one fluid going through this block. So whatever goes in minus whatever goes out must affect the change of the saturation of that fluid within that grid block. And we get an equation for oil and gas and water, and we've got other relationships that allow us to solve the system of equations to derive the pressures and the flows throughout the oil reservoir. Now what I'm interested in is something at the much smaller scale. I'm interested in modeling the flow between the sand grains. And this is something called pore network modeling. So what do we need? How are we going to model flow through this interconnected pore system? Well, we need some concept of a pore. What do we mean by that space between sand grains? We need some connections between the pores. We need some measure of what the shape of the pores are, and what a radius of a pore might be. We need a relationship that tells us what the flow rate through a pore will be if I put a certain pressure drop across it. We've already seen that perhaps we could use Poiseuille's law for that. We need some mechanistic understanding of how multiple fluids move through the pore space at this scale. So we're talking about tens of microns, fluids flowing through gaps of order tens of microns, very, very small spaces. And we need a big computer to try and solve all this. So the first thing to do is to take a very simple single tube and start sticking them together into an interconnected network. And so here I've taken 8 pores, and they're all different radii, and I've put them together in this sort of simple network. So these are just cylinders stuck together. I've got a big inlet here and a big outlet here, and I fix the inlet pressure to be P in and the outlet pressure to be P out, and I know what the size of each of these, what the radii of all of these different tubes are. And I want to solve the flow problem through that very simple network. And the way we do that is we look at a little equation that is conservation of mass equation at every junction, 1, 2, 3, and 4. So if we consider the flow at junction 1, and we're going to assume all of the flows always go into the junction, because we don't know, if you've got a big network, we can't assume that flow is going to be in one certain direction. So we have to assume that all of the flows are going into that node, and the mathematics will sort out the flow directions for us. So the flow in pore 1 going into node 1 will be pi r1 to the power 4 delta p, which in this case will be Pn minus P1 over 8 times the viscosity times the length of the tube. So we're assuming the viscosity is the same everywhere and all the lengths of the tubes are the same. And so if we do that, for each of the tubes going into node 1, we've got Q1 plus Q7 plus Q2 must equal 0. And we use Poiseuille's law and we stick that equation in to this top equation at node 1. So we've got Q1 plus Q2 plus Q7 equals 0. And if we put Poiseuille's law in there and we assume the viscosity is the same everywhere and the tubes all have the same length, we get this equation here. So we know what the radii are. We've got the pressure at node 1, P1, multiplied by a number, minus the pressure at node 2 multiplied by a different number, minus the pressure in node 3 multiplied by a number. Node 4 is not affected, it doesn't have any effect on that nodal flow, but we're going to keep it in there, you'll see why in a moment, equals a number, P in R1 to the 4th, and we know what P in and we know what R1 is, so that's a number. And we go to each junction and we do that in turn. So we get an equation at node 2, and at node 3, and at node 4. And so we've got 4 equations and we've got 4 unknowns. As we know from school, we can solve those simultaneously, and then we know what the pressures are at every junction in the network. And once we know the pressures everywhere, we can then go back to Poiseuille's law and calculate the elemental flows in every single tube. Okay, so big deal. But now we want to go up a scale. We want to start to study hundreds of thousands of pores or millions of pores so we can get up to a reasonable size piece of numerical rock, if you like. And so we do exactly the same thing. We take our big network and we set boundary conditions, an inlet and an outlet pressure. We conserve mass at every single junction. So that's what that is. The flows, what flows in must flow out, which is written in this form here. And we do that for every junction, so we get lots of simultaneous equations, hundreds of thousands of simultaneous equations, maybe millions of simultaneous equations. And we use linear solvers to solve those computationally. So we can design a rock where the red bits are these little tubes stuck together, if you want to think of it like that. They're all different sizes. We can use this technique to get the pressure everywhere. We then use Poiseuille's law to get the flow everywhere. And we can add all the flows coming out the end tubes, which gives us the total flow coming out of the network. So that's sort of the mathematical rationale behind what we're going to do. What about the physics? So the physics, this is one of Eric's old micromodels from the lab. So this is very, very old. So what we use to observe the physics is something called a micromodel. So I can pass this round. And if you look at it, you'll see an etched pattern of pores. So this is a flat glass plate that is then etched with a pore pattern that you're seeing here. And then a flat piece of glass is put on top so that the network is sandwiched between the glass. And you've got an inlet and an outlet port so you can start flooding fluids through these network models. And so what we're seeing here, we're looking down on it, and this is about 200 microns along here, so very small. This is glass. So this is the bit that hasn't been etched away by the acid. And these are the channels, and some of them are filled with pink oil, some of them are filled with blue water, and this is a white gas bubble. And if you notice, the interface between the gas and the oil is a curved interface. So at this scale, you get curved interfaces, and it's a bit like blowing a balloon up inside a maze. So as the pressures, we're going to drop the pressure in the micromodel, and the bubbles are going to start to expand, and they'll start to expand predominantly in the biggest parts of the pore space. So we set this going. So the gas starts to expand and it starts to displace oil, which then might displace a little bit of water. And this is what's going on in the middle of a reservoir. So everything's taking place pore by pore. So it's a very slow process in the middle of a reservoir. But by looking at these things in the glass micromodels, it gives us some rules that we can then use to develop algorithms to put in our simulator. So instead of just having a single fluid going through the network, which is what I told you about at the very start, we can now study multiple fluids going through the network. So this is more characteristic of what's going on in a real oil reservoir. And the last step, we need a big computer. When we first started, we were lucky if we could do sort of 10 cube networks using an old VAX computer. And then we got some of the very early Sun computers, and we got slightly bigger. And now you can run some really nice big models just on a laptop. So computing has just developed so rapidly that we can now start to model things at the actual physical scale. When it first started, you were simulating fluid flow on something that was a couple of millimeters cubed. And a lot of the interesting physics happens at a much larger scale, maybe the centimeter, tens of centimeter scale. And we don't just have to consider cylindrical capillary tubes stuck together now. There are now techniques where you can take thin sections, like use micro CT images, and build the rock so you have a numerical analog of the actual porous medium. So the pores now have a specific shape that is a reconstruction of the actual physical rock sample. And so that's some very simple way of building it. You can set up an Excel spreadsheet if you can use Excel, and you just set up a simple network model, and you can start calculating the flows through these things. But of course, we want to add more and more complexity. And so this is adding a little bit of complexity where now, instead of just having a rule where we've looked at those micromodels and we have a rule to say the gas fills the next biggest pore, then the next biggest pore, we actually want to calculate things numerically. And so now we have to include a viscous pressure field as well as these capillary forces. So those curved interfaces are like elastic interfaces that are stopping a fluid getting into the pore space. We have to include those sorts of that sort of physics and the viscous pressure driving gradients, the physics associated with that. We want to be able to track multiple menisci. We want to change the viscosity. We want to do a whole lot of more realistic things so that we can use our numerical model to look at some very interesting physics and then come up with hopefully some new ways of producing a reservoir. So here are some experimental results up here that were done in the late 80s in France. And this is injecting fluid that's a lot less viscous than the black fluid that's already in the model. So you're injecting from the bottom. And you're reducing the flow rate from left to right. And this is our model. This is Mohammed Regeg's model, ex-PhD student. And you can see that we broadly pick up the same sort of fingering phenomena. So we're not expected to match everything pore by pore because we don't know the actual pore system that was used in the original experiments. But we know approximately the flow rate and the viscosities and the average pore size. And you can see we reproduce the behavior quite nicely. The bottom set show a favorable viscosity ratio. So this is injecting a fluid that's much more viscous than the black fluid that's defending in the network. And now we get a piston-like advance, so we get a lot of the oil out of the system in this case, and not a great deal of oil out in this case. And so it's just showing that the combination of the curvatures of the interfaces and the rate at which you're doing an experiment and the variable viscosities, they all have an impact on how much oil you can get out of a system. And this is looking at something that's physically the size of one of these ceiling tiles, and it's a couple of centimeters thick. So this is a Bentheimer sandstone, and these are the experiments that were done in Norway by a chap called Arne Skauge and his group. And this is injecting water from the top and from the bottom. So these are two separate experiments. And you're injecting water into oil that's really viscous. So this is 7,000 times more viscous than water. And this is 2,000 times more viscous than water. So this is just like tar that you're trying to push out by injecting water. And you can see we haven't fiddled anything. We've just run our model. We've got lots and lots of pores in here. And again, we broadly pick out the same sort of patterns, the same sort of flooding behavior. Now, something interesting was observed that couldn't be explained at the time, is that when these fingers broke through, they started to swell, and you got some additional oil recovery as the fingers grew laterally. And this is exactly what we saw in the modeling as well. So we could explain this behavior. It was due to the balance of the viscous forces and the capillary forces at any given moment. And there's some recent micromodel confirmation of our predictions that have been done by a lab in the States just a few weeks ago. And this is sort of a cartoon example, but there are two PhD students working on this. I'm injecting surfactant at the bottom, so you'll see this go slightly lighter. There's a blob of oil surrounded by water and surfactants going in, and it's reducing the interfacial tension, it's reducing the forces that are holding that oil blob within the network. And as the surfactant concentration starts to build up, you'll see what happens when the capillaries on the top have their interfacial tension reduced. You start to flood out the oil. So this is important for something, for example, like low salinity water flooding, where you may be changing the contact angle and or the interfacial tension, or surfactant flooding. So we're interested in how these ganglia move, because when you pack your rig up and you've finished developing an oil field, you may leave maybe half to 2/3 of the oil behind. It's a very inefficient process. And so by adding surfactant or by changing the wettability, changing the contact angles, changing the curvatures of these interfaces, you could maybe start to mobilize these ganglia. And all of this is put together in this little package, numbskull. So this is a brief history of what we've been involved with over the last 28 years or so. Started with a very, very simple two-phase flow in water-wet systems. This is a small BP-sponsored project with myself and Ken. And we've added different physics, three-phase flow, unsteady state, depressurization. We've got a group looking at reactive flow in carbonates, Eric and Sebastian. Shale gas, Jing Sheng's interested in developing shale gas. So there's quite a lot of effort now. So it's been amazing. We could probably have filled this room with everybody in the world that was doing network modeling 28 years ago. And now there are many, many groups worldwide doing it. In current projects, still looking at low salinity water flooding. That's when you put low salinity brine to try and push the oil out. You can get different results compared to if you put high salinity brine in. Polymer flooding and ganglion mobilization. So that's going from tubes to interconnected network to rock. And now we want to take the knowledge that we've got from studying multiphase flow in rocks and transfer that somehow to flow in tissues and use that information to try and come up with novel treatment strategies for cancers, perhaps. So I'm going to concentrate initially on a process called tumor-induced angiogenesis. So there are broadly 3 phases of tumor growth. So we all have little tumors that are just sitting there quite happily in a sort of quasi-steady state. There are little bundles of cells in our bodies that are these sort of spheroids, little spheres. And in the center of that sphere is a necrotic core, just dead debris. And then on the outside, there's a proliferating rim of cells. So these cells are alive and they're dividing uncontrollably. And sandwiched in between, there's called a quiescent layer. So these cells are alive, but they're in a gap phase in the cell cycle, so they're not dividing. And so what happens is the proliferating rim starts to grow a little bit, and the cells that are close to the necrotic core can no longer get any oxygen from the surrounding vasculature, so they die. And so the whole thing just sort of slightly oscillates in a quasi-steady state way. So it just sits there and there's no problem. The problem comes when the balance of growth factors within the close to the tumor, that balance starts to be disrupted. And if that balance gets disrupted, maybe smoking or ultraviolet light or something that's affecting the balance of chemicals, you can start to create new blood vessels. So this is a tissue. This is a parent blood vessel running around the edge here. This is a pellet that's got a growth factor associated with it, and that growth factor is diffusing through the tissue. And what happens is it causes these blood vessels to rupture, and new blood vessels grow towards up gradients of this growth factor. So this is why cancer is called cancer, because these blood vessels look like the legs of a crab. So we've got lots of benign tumors and it just sits there, but the bad news happens when you start to get angiogenesis and vascularization, because then the tumor gets more nutrients and it starts to grow more rapidly. And then eventually, at a later stage, parts of this tumor can break off, go into the bloodstream, go to other sites in your body and lodge elsewhere, and then it's the secondaries that are usually really bad news. Now, angiogenesis as I mentioned earlier, is the formation of these new blood vessels. And it plays a vital role in everything biologically to do with tissue. So in tumour progression, it's not a very good thing. In wound healing, it is quite a good thing. You want to get some oxygen to your wound area to help it heal. So you've all got cuts, and if you rub your cut, I've got a lovely cut across the top of my finger. If you rub it, it just turns white, just neat collagen at the top, and the blood vessels haven't infiltrated that wound. So it's a good thing for wound healing. And in retinal development, you want some of it, but you don't want too much of it. So if you get macular degeneration, you get lots of overgrowths of capillaries that can lead to blindness. And so by understanding how these capillaries grow, we can start to maybe look at things to stop them, the capillaries growing, anti-angiogenic treatments. So this was the great hope for cancer therapy, to give an anti-angiogenic treatment. If you could kill the capillaries off around a tumour, you could starve it of nutrients, and that would kill the cancer. It turned out not to be as straightforward as that, but that was a very hot research area for a while. Radiotherapy depends upon the local oxygen concentration, so that's affected by perfusion, by the blood flow. And maybe we want to deliberately create angiogenesis. If you had a heart attack, you might want to deliberately try and grow blood vessels into the scar tissue, around the scar tissue. And flow plays a vital role in determining the architecture of these capillary beds and how they adapt. So tumor-induced angiogenesis at the cellular scale is something like this. This is the sort of the cartoon story of it. You've got your tumor that's hypoxic, so it's got low oxygen concentration, and it starts to recruit new blood vessels from the pre-existing blood vessels. So you've got hypoxic tumor cells and TAMs, tumor-associated macrophages, little white blood cells that also send off a signal to say, right, we need to get some oxygen here, and they secrete tumor angiogenic factors, or TAFs. And the endothelial cells start to eat away, and they start to tunnel through the release of protease as they migrate towards the tumor. So these MMPs are matrix metalloproteinases. They're little chemicals that the tip cells of growing capillaries secrete, and they start to eat away at the tissue as they burrow through. So it's a bit like a wound healing response that never heals. Once the tumor is supplied with nutrients, you get accelerated growth, and then you could have these fragments that snap off and go to different sites in the body and give you secondaries. And this is an experiment showing a implanted tumor fragment, and here are the existing vessels, and you can see that over time, over 20 days, you get this tumor growing really quite rapidly. So to the uninitiated, the next slide's not very pleasant to look at. So it's this one here. So this isn't my model. This is the model of Sandy Anderson and Mark Chaplin. So they came up with this way back in 1998. And it's a model that tries to capture that process. And it's called a hybrid model because you've got a continuous equations dealing with the chemistry, with all the chemical signals and the proteins. But you discretize this equation, which is the equation that tells you how the endothelial cells, these are the cells that make up the capillaries, how they move through the tissue. And so what these endothelial cells do, there's some random motility. So cells don't just sit there, they're constantly throwing out little feet to try and sense their environment called filopodia. So they're constantly on the go in some sort of random motion. They also want to climb up gradients in angiogenic factors. So if they see a chemical gradient, they start to move up it. And there's also stickiness associated with the proteins in the tissue. So if you've got any gradient in the stickiness, so say fibronectin or vitronectin, which are proteins in the tissue, that holds the capillaries back. So they also want to climb up gradients of fibronectin. So that's what these terms mean. There's a diffusion coefficient multiplied by some random motility. There's a chemotaxis term. This is the gradient in growth factor interacting with endothelial cell density. And this is the gradient in protein density interacting with the endothelial cells. This equation tells us that the growth factor itself is taken up by the cells. So the tip cell, Ni, so it's taken up. So this is decreasing as the capillaries grow in the tissue. The fibronectin is being produced by these tip cells and is being degraded by this M, which is the protease, the matrix metalloproteinase, which is produced by the tip cells. It diffuses, and there's some natural decay. So you've got these continuum equations in the white box, and this equation you discretize, and you end up with some movement probabilities for some sprout tips. So it'll give you some weightings as to which direction the sprout tip should move in response to the local chemical environment. And then there's branching. There are rules for branching, and if blood vessels join together, then you have to kill one of the tips and just let one of them go, because these blood vessels fuse together to form loops which can then flow. So this is what I'm simulating here. So this is the parent vessel across the top. I've just started to sprout 5 tips, and this is the concentration of the enzyme that's eating away at the tissue. So blue's low concentration and red is high concentration. And as we continue with the simulation, the capillaries grow towards the tumour surface at the bottom. So there's a gradient of growth factor, high here and low here, and they branch and they start to feed the tumour. So that was the model that Sandy and Mark had come up with. And when I bumped into Jonathan, it was literally picking up one of these newsletters at Heriot-Watt, which a long time ago used to be physically on pieces of paper, not on the internet. And I picked it up, and Jonathan just was interested in speaking to people who were modelling things at the microscopic scale. He was interested in wound healing and cell migration. And I contacted him, and he put me in touch with these guys in Dundee. And so I coded up their model, and I took it up and said, this would be really quite good if we could flow it. So we got together and we decided that we could take their patterns and we could plug their patterns, overlay it onto my flowing template that I've been using for the oil work. And so we can then, just as we did at the very start, so again, it's just the simple tubes again, we can start to solve the flow problem through these interconnected capillary networks, but now we're talking about blood capillaries, not the holes in rocks. So this is what we did. This is the first thing we did. We injected a drug, or really just a tracer. It wasn't doing anything. It was just a tracer at the left-hand side of the parent vessel at the top. And we tracked it. We had a tracer algorithm that allowed us to track the tracer concentration as it flowed through the network. So red's high concentration and blue's very low concentration. And you can see that what we found was we got a lot of bypassing. The drug came in, went back to the parent vessel, and didn't get anywhere near the bottom of the tumour. And in the very first paper we came out with, we just did some very simple things. We changed the viscosity, we changed the radius, and some very obvious things came out. So this is treating the blood as a simple fluid like water, Newtonian fluid. But unfortunately, blood's not a Newtonian fluid. It's quite a complex fluid. So here are some red blood cells flowing through branched capillaries, and you can see that it's unsteady. It's not a continuum. You've got these individual blood cells that are shaped like parachutes. They're trying to get in there. Their diameter is about 8 microns, and the capillary is obviously a little bit smaller, so they have to bend, they have to change their shape to get through there. And also, the whole bed's not static. In very short time scales, okay, in a rock things can change over quite a long period maybe, the whole bed can start to shut down. So you might get a metabolic signal and all the capillaries are starting to shut down. As we know, if we exercise, if we get cold, capillaries start to dilate or constrict. And so we wanted to put this into a more realistic model for blood flow. And lucky for us, there'd been a group in Berlin that had done some experiments in the late 90s. They'd taken something called a rat mesentery. So that's almost a 2D sheet from a rat that has got a nice branch network of blood vessels, and they could flow it, and they could measure the flows, and they could history match it. They could decide, they could choose a formulation for sort of an apparent viscosity that somehow captured this complex motion of individual blood cells. So we could treat blood as almost a continuum fluid like water, but it captured the biphasic nature of blood. And so this is telling us that the viscosity, the apparent viscosity of blood, depends upon the fraction of red blood cells that are in that particular capillary, but also the radius of the capillary itself. So although this looks quite a complex equation, it's just giving me a relative viscosity as a function of how many red blood cells I've got in each capillary and how big the capillary is. The fact that these vessels are not static but they're constricting and dilating needs to be put into the model. And again, this group in Berlin had come up with a model that told us how these radii would adapt depending upon a lot of different stimuli. So there's a wall shear stress, so depending upon kind of the friction at the wall, the surface of the capillary may dilate or constrict. There's a transmural pressure term, so depending upon the pressure difference within and without the capillary. There's a metabolic stimulus, so these endothelial cells that are forming the capillaries are somehow giving off some chemical that's flowing downstream and affecting the radius of downstream capillaries. And a shrinking tendency, so this whole equation doesn't blow up. And so we put that into our model. So now you've got to iterate a solution. You can't just solve it straight away, because now you're flowing it a bit. The radius is dilated a bit based on the previous equation. That's affected the viscosity, which has affected the pressure solution, which has affected the flow, which has affected blah, blah, blah. So you have to iterate a solution. So this is growing the network. So green are very thin capillaries and red are very thick capillaries. These have dilated. So we start off, the capillaries all begin very thin, constricted, and then we put the previous 2 slides worth of maths into the model and we flow it, and we start to get this dilated pathway shown in red. So at the end, this is the blood vessel network we've got. We've got some little capillaries in green, some medium-sized capillaries in light green, and some really dilated capillaries in red. And what it shows is that you get shunting behavior associated with sort of tumour vasculatures. So if I put my drug in here, it's all just going to flow through this shunt and come straight out the end again. It's even worse than it was when I just assumed a Newtonian fluid. And because we've got a numerical model, we can change all the parameters and see what impact that has on the vessel architecture. So we can change things like the haptotactic coefficient. So how much does the sticky gradient affect the growth of capillaries? What about the plasma viscosity or the base level of hematocrit? So we can look at this, but you can see you get different vasculatures, but they all exhibit this shunting behavior. And so you can go to the literature and you can look a lot of these histological sections out of tumour vasculatures, and it's fairly easy to find something that looks as messy as this. So it's not a great test of the model. So you get a messy vasculature, you get shunting, and you can find a lot of pictures that look similar. So it's not a good test of the angiogenesis model. So is there a better test of the angiogenesis model? Now say that as if there's a nice linear story here. Of course, this isn't how it works. You do this and you're very pleased with it, and then you go off, then you get a project looking at the retina, and it's sort of, it's a useful, it's a more stringent test of your modeling protocol compared to this in hindsight. You didn't know that at the time. So let's talk about retinal development now. So the mouse retina is what we considered in our joint project with the researchers at Culrain. So the retina is an amazing system. So metabolically, it uses loads of oxygen, considering the size of it. It needs lots and lots of oxygen to stay healthy. And it's kind of a 2D system. You can look at the surface of it. Your optician can see if there's anything wrong with you. It's a good prognosticator. And it's made up of these alternating petals of arterial and venous blood. So it's a beautiful structure. And this is panning through the top. When I set this going, this is panning through the retina. So you see the top surface, you've got these nice blood vessel trees. And as we pan down, you'll see dots. And these are perpendicular capillaries going into a second layer. And then there's a third layer underneath. And this top layer is really, really beautiful, and it's a real test of modeling. Can you reproduce this sort of patterning? It's a bit like a tree leaf. Can you reproduce these alternating petals? So the underlying cell biology is quite interesting. So initially you've got your optic nerve, and these nerve cells come out, these retinal ganglion cells, and they secrete a growth factor called PDGF alpha, and they start to drag along cells called astrocytes. So these, as the name suggests, are star-shaped cells, so they form a big interconnected web. And when these become hypoxic, so they're moving away from the optic nerve, so they're losing contact with an oxygen supply, they start to secrete VEGF, another growth factor, that then pulls along the endothelial cells. So you get this wave of astrocytes followed by a wave of capillary cells. But it's not remodeled yet, it's just a big plexus of endothelial cells. And then eventually you get cells called pericytes, that sort of little muscle cells that grab onto this blood vessel network and stabilize it. And so this is looking at the retina developing in the mouse just after it's born, up to 16 days after it's born. So you see, you start off with just a mess, all the astrocytes and just a mess of endothelial cells, and eventually it evolves to this leaf-like structure. And so we thought, let's put this into a mathematical model. And so now, instead of the tumour where we had high growth factor in the middle of the tumour bringing vessels towards it, we've now got an optic nerve and we want vessels to grow away from it. So we set up this PDGF alpha gradient. So this is pre-secreted by the astrocytes. We set that up. And then we have astrocytes coming from the centre of the system responding to that gradient. And then when they become starved of oxygen, they are going to secrete VEGF, and that's going to start to drive capillary cells from the optic nerve to the outer reaches of the retina. As I say, we've got these 10 alternating petals of arterial and venous flow. In this model at this stage, there's no flow at all. We're just looking at the pattern. So we get a set of equations for the astrocytes that are very similar to the ones I showed before for the endothelial cells. We've got endothelial cell equations, and again, we've got branching and anastomosis. And we can code these up and start to run the simulation. So this is what we get. So this is embryonic day 19.3, so it hasn't been born yet. And these are the astrocytes that have come out, and they're moving out from the optic nerve, creating in a radial fashion. They're secreting the growth factor that is now drawing out the purple capillary cells. And so you've got these two areas of interconnected cells. And we can match the experimental data. We've got tuning parameters. We can match the experimental data from the embryonic stage right through to late stage after birth. And this is the curve, the simulated curve of astrocytes in blue and the endothelial cells in purple, and the experimental data are in black. So these are choosing sensible parameters. We're not choosing mad things for our model, and the model can reproduce the data quite happily. And we can change things like the diffusion coefficient to look at different isoforms of this growth factor to try and predict what might happen to the developing retina. How will the blood vessels form or be transformed if we use a different growth factor? And so you can do this experimentally and study what happens to the retina and then try and uncover what might be going wrong in certain pathological conditions. So there's no flow yet. And because I had a very basic, simple model of flowing from the left to the right, I saw this as being topologically quite tricky to try and flow this system. And so what I did was I separated the whole thing into three layers. I had a high pressure arterial blood system, which is the red one here. So this is a little manifold of tubes in red coming from the left inlet to the middle optic nerve on one layer. Then a layer behind that, from the outlet going towards the optic nerve, I had a low pressure blood system, the venous sink, which is this blue section here. And they were firing, there were pipes coming perpendicular to those manifolds coming out at you towards the surface here of the cube, which is the retinal surface. So you've got this high pressure system, a low pressure system behind it. They're firing alternating tubes up to the surface, but nothing's connected yet, so nothing's flowing. They're just sitting there ready to flow if a connection should be made. And so then we run the angiogenesis model, the retina model on this top surface, so that now we're going to start getting connections between the high pressure arterial system and the low pressure venous system. So once these tubes start to get connected, blood will flow from the left up to the retinal surface, around the vasculature, back down to the low pressure manifold, and then out the right hand side. So we thought this was going to be an absolute cinch. And this is what we got. We got a huge shunt forming around the optic nerves, these massive mess of capillaries, really highly dilated capillaries around the optic nerve. And the hematocrit, so the red blood cell concentration, didn't get anywhere near the outer reaches of the retina. the developing retina. And we had a very miserable time in Belfast Airport, which is not the brightest place at the best of times, but we were wondering what on earth was missing in this model. Was it pattern formation? What we were sort of grasping at straws here. And then my then PhD student, Mike Watson, found these extra stimuli that had been, again, written down by the group in Berlin, had been forgotten about for a long time, but then they were republished in about 2010 in Cancer Reviews. And so these extra stimuli we could then put into our model. So these stimuli, one of them is a metabolic stimulus, so every part of the blood vessel network is giving out a signal that's affecting everything downstream of it. And there's an electrical signal that's propagated against the flow that is dropping off exponentially, that's also affecting the constriction or dilation of the capillaries upstream of it. And there's also something called the Porous effect, so that when blood flows to a junction, you can't just split the red blood cells in proportion to the downstream flow. There's something called plasma skimming, where the red blood cells sit in the middle of a capillary and the outside of the capillaries just got neat plasma. And so there's a flow law that tells you how to distribute the red blood cells as you flow down past a junction. So we put this in. So Mike just came up with this very simple network. We're coming in here and we're going out here, and you can see the whole thing gets dilated. The red is the really thick capillaries in the old model. And when we substitute that with the new model, we get this. We get some big capillaries at the outlet, some reasonably big capillaries at the inlet, some medium-sized ones here, but the nice thing is we get some small capillaries far away from the inlet and the outlet. So this looked promising. And so we put this into the model and we set it running. And this is what happened. So the top of the blood vessels and the bottom is the concentration of red blood cells. So we grow it and then we flow it for a bit. We grow it and we flow it. And you can see that the alternating petal structure emerges completely naturally from putting those two stimuli in. And that has implications for the tumour, because if the tumour doesn't have this, it has shunting. It suggests that the tumour biology is missing these two stimuli, because this is what's keeping everything nice and making everything a nice pattern developmentally. So this is looking at the vasculature, the tissue oxygen concentration, the red blood cell concentration, and the vessel oxygen concentration. And this is a comparison with the mouse data. So we didn't try to fix anything here. These are just slices from the model versus the model versus the actual experimental data, model experiment, model experiment. So you see we reproduced qualitatively the features quite nicely. So what about current challenges and what we're going to be looking at in the future? Well, the first thing we're looking at is coupling the angiogenesis model to a more sophisticated model for the tumour. So we did this way back in 2009 with a group at the University of California at Irvine, and they had a nice model for the tumour growth using level set methods to track the surface of the tumour, and we had our angiogenesis model, so the idea was to couple these. And so we're looking at here at what's going on in the tissue. You've got this brown dead region, the blue quiescent region, and the red proliferating rim of the tumour. You've got the hematocrit. There isn't any yet because the blood vessels haven't grown. The oxygen concentration, so we've got some oxygen diffusing out the top parent vessel, but not a great deal of oxygen below it. This is the stress field, the pressure within the tissue, the vessel radii, and the TAF concentration. So we've got a hypoxic tumour, so you've got a high concentration of the growth factor. It's crying out for angiogenesis, and a low concentration here. So you've got gradients in TAF. And so if we set this going, you'll see what happens. The blood vessels contact the tumour, which makes it grow, but the tumour itself starts to compress some of the capillaries, so the oxygen delivery to that region decreases. So it becomes hypoxic, so it secretes more VEGF, so it attracts more capillaries, and so the whole thing goes round in this cyclic fashion, and your initial nice bundle of spherical bundle of cells becomes very disordered. So what we want to do now is we want to extend this. I'm just going to keep playing. We want to extend this to couple it to a cell-based model. So Cicely McNamara and Mark Chaplin at St Andrews are working on this individual-based model. So this is a tumour growing, and the cells are flashing when they're dividing. So there's a lot of biochemistry and a lot of maths involved in just that ball of cells growing. And we've put it together with our blood vessel network. And the idea now is to try and track fragments that are breaking off. When are you more likely to have metastases breaking off, going to the bloodstream and giving you secondaries? So this is part of a big project with Glasgow and St Andrews and Sheffield called SoftMech, soft tissue mechanics. In a few weeks' time, I'm going to Glasgow to talk to some clinicians about how we could maybe apply the retinal development model, because there are some interesting things to look at here. One, for example, retinopathy of prematurity. If you've got a premature baby, tend to be given hyperbaric oxygen to help them develop. Unfortunately, that doesn't allow the retina to develop very well, because as I showed you, the retina needs to be starved of oxygen a little bit to get these blood vessels growing. And so premature babies can often become blind if they're given hyperbaric oxygen. And so there are various things to look at. Sickle cell anemia, again, is looking at the changes in viscosity as you change the cell shape. Diabetic retinopathy and age-related macular degeneration. So there are lots of things to look at with this model. We've also looked at modeling drug delivery to glioblastomas. This is work that we did with Harvard. This network here was taken from a real mouse tumor, and we came up with some flow equations that allow us to track tracer going into that network, spilling out, extravasating into the brain tissue, and trying to see what impact different parameters would have on what you would see in the imaging. So we can change parameters in the model, and some quite surprising things happen. For example, if it's a high flow rate and a high value for tissue diffusion coefficient, you would expect maybe a lot of the tracer, a lot of the drug, to leak out into the tissue and spread everywhere. What seems to happen is, under those conditions, the drug does leak out, but it gets quickly reabsorbed by the downstream vasculature. So it doesn't light up under MRI. You'd expect to see a nice bright blob of contrast agent under MRI if you had a big tumor, but there are conditions where it won't light up. So it's been working with them to try and pick this apart bit by bit. Looking ahead, just to finish with two quick examples, we're interested in with Glasgow looking at a pulsating network. We're interested in what happens after a heart attack, so we want to look at angiogenesis post-myocardial infarction. So we've got the whole network responding to an external heart stimulus. And microbubble transport. So this is work that I've been doing with Vasilis over there and Vaso. This has implications for contrast-enhanced ultrasound. And interestingly, it can also be used for looking at fines migration through porous media, so it completes the loop back to petroleum engineering. So I'll end with two slides on contrast-enhanced ultrasound, and Vasilis can put me right if I'm talking absolute rubbish, because I'm just learning a little bit about this. So it's a well-established, it's very safe, and it's very portable, and it's far cheaper than other imaging techniques. But the image quality is often quite poor, and there are big uncertainties associated with it, and there's often operator error as well. And so there's a possibility of using microbubbles. So it's already used in looking at liver cancers, where you could put a lot of microbubbles into the liver and see where dark patches appear, and that corresponds to tumors in the liver. But it'd be nice to track individual bubbles, and this is what Vasilis in his group can do. They can track individual microbubbles to try and reconstruct the underlying network of capillaries. So the clinical aim here is to reduce the number of biopsies you might need. So instead of taking lots of biopsies, you could use an ultrasound probe and investigate what the underlying structure is to better stage the cancer and better assess what treatment you're having. So here's some very simple examples that Vaso's produced. These are you might be able to see little yellow bubbles going into this branch network. And then these bubble positions, we know exactly where they are. We can send that file to Vaso, and she and Vasilis's group can produce an artificial ultrasound image of those bubbles going through the network. So why on earth would you want to do that? Well, when you've got more complex interconnected capillary networks, you would like to know the structure at different stages of cancer development. And so it's quite hard to see here. It's quite hard to see here, but there's an angiogenic network here, a messy network like I showed in the earlier slides. And this is the ultrasound image that's been produced. And so what Vaso's doing at the moment is she's constructing an artificial prostate vasculature and starting to blast out certain areas of that vasculature to represent tumours in different places of the prostate and producing artificial ultrasound pictures. And then the hope is to use machine learning to come up with a biomarker that tells us what stage the cancer's at. And this is all put together in a numerical model NumT. So this is a little user interface that you can start to play around with. You can load in the mouse network and give various treatments, etc. So I'll finish with a couple of conclusions. So I hope I've been able to demonstrate that mathematical modeling can give a great deal of insight into flow problems at the microscopic scale. In different research areas, we've gone from a simple tube, remember, to an interconnected network of 8 tubes to try to model flow through quite complex rock systems. These models are useful in explaining sort of contradictory behavior in the lab. So you could get different labs doing what seem to be very similar experiments but coming up with very different conclusions. And you can use these models and run lots and lots of numerical experiments to try and uncover why these contradictions might exist. And we can also use them to suggest novel ways of increasing oil production, making it more efficient, but also on the green side of things to help with CO2 storage solutions. So we can look at how carbon dioxide migrates through rocks at this scale, for example. My blood flow results have highlighted a number of important targets for therapeutic intervention, which I haven't really talked about here, but you could use these models to target different parts of the vascular network. You might want to target immature vessels, that's what an anti-angiogenic treatment would do, or use antivascular agents to target more mature parts of the blood vessel network. I would finish by saying I think some of the most challenging but also most rewarding opportunities are at the interface between disciplines. But the problems are there. There's a communication barrier often trying to speak to biologists trying to speak to mathematicians. You know, there are 17,500 three-letter acronyms, and I think the biologists use about 17,487 of them. Similarly, we have to try and tell them where modeling might be useful and what the model cannot achieve. And finally, I would say that I think there's great benefits to be had by trying to apply conventional modeling techniques in an unconventional setting. So I'll finish there. Thank you very much.